to talk to you today about CLL in 2023 and the future ahead. These are my disclosures. So I'm going to briefly touch base on the frontline CLL, which probably you heard my talk about the combination strategy just before this. So I'm not going to spend too much time on frontline CLL because I covered in that talk. I'm going to talk mostly of the relapse CLL. But in the frontline CLL, I think I showed this slide in the previous session as well. I think it's really important to see the development which has occurred in the field of CLL with so many new drugs, and we have moved away from chemotherapy for these patients. Now, when you look at the frontline therapies, target therapies in the frontline CLL, again, there are 4 of these therapies which are approved: ibrutinib, acalabrutinib, zanubrutinib, and obinutuzumab, which I discussed in my previous talk. But I think when you look at the future ahead for frontline therapy, I think, and this is something we discussed in the previous talk, I think the BTK inhibitors, namely ibrutinib, acala, or zanu, and you can also argue about pirtobrutinib here, plus BCL2 inhibitor. Obviously the main one is venetoclax, but there are other BCL2 inhibitors in clinical development, including planned to be going into phase 3 studies very soon, where, and depending on the phase 3 results, may get eventually approved, with and without the CD20 antibody for obinutuzumab. And I think this is a very important strategy, which again is being tested in many different clinical trials, either phase 2, phase 3. And I think in the next couple of years, we'll know more which of these regimens are going to make it to an FDA approved list, to say, and then we maybe start using them more and more commonly for our patients. So I'm myself looking forward to data with 2nd generation and 3rd generation BTK inhibitor in combination with the BCL2 with and without CD20 antibody. And I think that will really provide a time-limited approach for our patients, maybe without the need for IV infusions of obinutuzumab. But again, the time will tell, depending on how the phase 3 data reads out. I'm going to focus most of my second talk in the relapse CLL and kind of give you some concepts of where we are heading there and what are the kind of unmet needs in the relapse CLL phase. So when you talk about relapse CLL, you have to think about what are the regimens we're talking about. So certainly the same drugs we talked about previously, ibrutinib, acalabrutinib, and zanubrutinib, are FDA approved in relapse CLL. Pirtobrutinib is not yet FDA approved for relapse CLL, but at least in the United States, we are able to access to it because it's on the NCCN guidelines. And again, this drug may be available to you as well in your country, depending on if it is already available for mantle cell lymphoma. But again, the expectation is that this will get approved for CLL down the line. For BCL2, we only really have venetoclax right now, but there are at least a couple of other BCL2 inhibitor in phase 3 studies. PI3 kinase inhibitor, I'm not really going to talk about them. They have kind of fallen out of favor in terms of the toxicity profile and efficacy, but certainly can be used if you're running out of options. And in terms of the CD20 antibody, the preferred agent for CLL is obinutuzumab, though certainly rituximab could also be used if obinutuzumab is expensive or not available. Now, when we talk about relapse CLL, one of the important aspects of the treatment choose in relapse CLL is basically depends on what the patient has received previously. And you can think of several scenarios, sequencing scenarios for these patients. For example, the most common scenario currently plays out is a scenario number A, where a patient is on a covalent BTK inhibitor, for example, ibrutinib or acala or maybe zanubrutinib. They get it for 7-8 years, then they relapse while taking the drug. At that time, you switch them to venetoclax plus CD20 antibody for a 2-year regimen. Then you enter this treatment-free remission where the patient is not taking any drug. And when they relapse, then you can use a non-covalent BTK inhibitor, which will be like pirtobrutinib, or there's another one in development called nemtabrutinib. You can retreat them with venetoclax, or you can do CAR T-cell therapy or look at other potential clinical trials, including, PI3 kinase inhibitors or other drugs. So that's, I think, the most common scenario kind of which plays out. Or you can have a scenario where you start with ven obinutuzumab for a 1-year therapy, a time-limited approach. You get 6-7 years out of it. Then patient relapses. You at that time add a covalent BTK inhibitor. You get many years out of it, and then you use your next line of therapy, including repeating venetoclax. You could do that. Or you can start with venetoclax obinutuzumab. You retreat with venetoclax obinutuzumab, and in the third line, you introduce your BTK inhibitor. Or could you envision a scenario where you are starting with a covalent BTK inhibitor? When the patient progresses, you switch them to a non-covalent BTK inhibitor. So you're staying in the same class. You're not going to venetoclax yet, and then you're using venetoclax in the third line setting. So there is no way to know which is the right strategy here. And I think there's also other regimens or sequencing scenarios you can think of, but these were the common ones I thought I would highlight. Important point here I'm trying to make is that if you look at the x-axis, we're talking in the years, like 15 plus years. So that's the positive thing for our patients with CLL, is that these patients are living longer with the therapies we have, which also makes the designing trials of comparing these regimens very difficult of the time and commitment it will take to run those trials. But I think it's all a very positive news for patients with CLL, seeing such long-term remissions achieved with these drugs. Now let's kind of look at some data, which is very important to about these BTK inhibitors in the lab setting. We have 3 BTK inhibitors: ibrutinib, acalabrutinib, and zanubrutinib. All 3 are approved for CLL. This slide shows that they had different targets besides BTK. So certainly BTK is the main target they inhibit, but you can see ibrutinib inhibits several other targets, acalabrutinib a bit less, zanubrutinib maybe kind of in the middle. So they have different off-target effects, which can potentially explain some of the side effects. Now, I wanted to highlight 2 different trials, which are very important trials in the field, which came along in the last couple of years. The first trial compared head-to-head acalabrutinib with ibrutinib for relapsed CLL. And this was a trial which was designed for del17p, del11q patients. So these are patients who have high-risk genomics, and they got acala versus ibrutinib. And there was no C20 antibody involved, and the primary endpoint was non-inferiority of the progression-free survival. So this is the long-term follow-up, the most recent one. The median follow-up is about 40 months. And as you can see here, the curves are kind of overlapping. Maybe at early point of the curve, acalabrutinib was kind of superior, but then later part, they are kind of merging together. But it met the primary endpoint that acalabrutinib is not inferior to ibrutinib. Importantly, when you look at the side effects, whether it's atrial fibrillation, bleeding events, hypertension, they were more frequent with ibrutinib compared to acalabrutinib. And this is also shown kind of graphically here, where AFib and hypertension are much more common with ibrutinib than acalabrutinib. And then other aspects of bleeding, diarrhea, arthralgia also less common. So at least in this trial, we see that safety is much better with acalabrutinib and efficacy appears to be equal. We also saw, this was at the ASH meeting and then subsequently published in New England Journal, zanubrutinib data. And the zanubrutinib trial is called Alpine trial, where patients were again randomized in relapsed refractory to zanu versus ibrutinib. With the caveat here, is important distinction here is that this enrolled no high-risk genomics were not selected. So any relapsed refractory patient was allowed to be enrolled on the study. And in this study, at the 2-year follow-up, they saw a statistically significant improvement in PFS compared to ibrutinib. And this was probably the reason it made it to the late breaker and in the journal that zanubrutinib is statistically superior to ibrutinib for PFS at 2-year mark. Now, I guess it remains to be seen what will happen to these curves with the longer follow-up. As I mentioned before, with acalabrutinib plus ibrutinib, early part there was a separation in favor of, appears to be separation in favor of acalabrutinib, but at 3-4-year mark, the curves overlapped. So I guess I'm looking forward to a longer follow-up of this data to see what happens at the 3 and then the 4-year mark. If you look at the AE profile in this trial, actually, Zanu versus ibrutinib, at least for the numerically, the side effects appear to be quite similar, except atrial fibrillation, which was lower with zanubrutinib than with ibrutinib. So certainly these two trials have, made, I think it's quite clear that the second generation BTK inhibitor, namely acalabrutinib and zanubrutinib, are safer than ibrutinib, and therefore, at least in the United States, I think, and that should be the case elsewhere, we have preferentially started using second generation BTK inhibitors for a patient as opposed to ibrutinib. Now the question comes up, which should I pick, whether acalabrutinib or zanubrutinib? I think with the data we have, I don't think there's a clear-cut winner. These drugs have not been combined head-to-head, will never likely be combined head-to-head. So at the same time, I think it just depends on your own clinical preference for patients, maybe cost, maybe consideration as well. But otherwise, I don't think there's a clear-cut reason to pick one or the other among the second generation BTK inhibitors. Now, that was the BTK inhibitors. And I want to introduce a concept of an unmet medical need, which is kind of emerging in the context of CLL. So we routinely kind of hear about the, obviously there is so much positive development in the field of CLL, and that's all exciting. But at least in the clinical practice, I and I'm sure many of my colleagues are starting to see these patients who are being termed as double exposed or double refractory. So what is a double exposed patient? Double exposed patient means patient who has a prior BTK use or BCL2 use, and they have stopped them for whatever reason. It could be toxicity, it could be progression, it could be any reason. So at least they have received those drugs. And the double refractory is a definition in evolution, I will say. I think there is no one accepted definition, but for BTK inhibitor, it's kind of easy if you progress while you're taking a BTK inhibitor, and you progress while taking a BCL2 inhibitor, or you took a BCL2 inhibitor, you stopped it, and then within 1 to 2 years, you have progressed. But again, this definition of progression when you start BCL2 inhibitor is not a uniform definition. But nonetheless, these are the group of patients which we are starting to see more and more in clinical practice, because obviously both BCL2 inhibitor, namely venetoclax, and BTK inhibitors are now approved for several years, and patients then are failing one, they go to another one, and then they're failing that, and then they're coming to clinical practice or refer to us for clinical trial options or other options. So that becomes a challenging group of patients kind of to treat. Now I want to highlight to you two new strategies which are being pursued for kind of this group of patients. These are the group of patients, double refractory, double exposed, where they have had a covalent BTK inhibitor, ibrutinib, acalabrutinib, or zanubrutinib, and also had venetoclax in the past. So one such agent, which I think has shown quite potent activity, is pirtobrutinib. Pirtobrutinib is a third generation BTK inhibitor, but unlike the other BTK inhibitors, covalent BTK inhibitors, pirtobrutinib, first of all, is very, very specific for BTK. And secondly, it works for patients who had a cysteine 481S mutation. So that's the resistance mutations to covalent BTK inhibitors. So this drug, I'm sure you have seen this data. It has been presented several times at the ASH meetings and other meetings. And the most recent update was from this past ASH, where they treated 261 patients, or they reported on 261 patients with CLL, all of whom had prior BTK inhibitor, and 41% had prior venetoclax as well. And then the BTK inhibitor they had, 3/4 had progression on BTK, remaining 1/4 had a toxicity to BTK. If you look at these patients, 3 prior lines of therapy, so quite heavily pretreated patient population. And when you look at the responses, this is the lymph node response rate, and you can see quite dramatic responses in the lymph nodes for these patients. And the dark blue color are the patients who had actually discontinued BTK, for example, ibrutinib, because of progression. And you can see that practically most of the patients are, really all the patients are responding. So I think this drug is actually quite an important drug in the field and leading to quite nice remissions for patients. Now, when you look at this data, and this data has now matured, When you look at all comers, all 200 plus patients on the study who got 3 prior lines of therapy, the median progression-free survival is about 20 months, so just under 2 years. And if you specifically look at a group of patients who had both BTK and BCL2, so these are exposed patients, had a BTK exposure and a BCL2 exposure, median prior lines of therapy is 5, so quite heavily treated patients. The median remission, median PFS was about 17 months. So I think an important point here is that these drugs, pirtobrutinib, is actually quite effective. It controls the disease well, leads to partial remission in the majority of the patients, but you're seeing these progressions over time. So hopefully, when this drug is introduced in earlier lines of therapy, we will see a higher rates of remission duration, but at least it's able to get to remission majority of the patients. But then you start thinking about what I'm going to do next for my patients, because these remissions are likely not going to be long-term remissions. Now, another very important field which is emerging in the context of CLL is the BTK resistance. So you have probably heard in previous talks that BTK cysteine 481S mutation is a resistance mutation, which is described with ibrutinib and zanubrutinib and acalabrutinib. And that's well described, and there are assays which are available you can run, and we are able to do it here as well in our lab to check for resistance mutations to BTK. And again, really all the data, previous literature was with cysteine 481S. That's the resistance mutations to BTK, covalent BTK inhibitor. Now, last year, the group from Sloan Kettering reported on the pirtobrutinib resistance. And they had 9 patients who had pirtobrutinib, and then they failed pirtobrutinib, and they had a pre and a post treatment sample. And the main message of the paper was that 7 of the 9 patients acquired a non-cysteine 481S mutation. So these are mutations not in the cysteine 481S, but other nucleotides of BTK protein. So these are the 9 patients, and I just want to highlight a couple of these patients for you where, for example, this particular patient had this PLC gamma 2 mutation. He got pirtobrutinib, he or she got pirtobrutinib. When the patient relapses, PLC gamma 2 mutation is still present, but they have acquired this BTK L528W mutation. This other patient had a cysteine 481S mutation. When they progress, cysteine 481S mutation is not to be found, but they have developed a BTK L528W mutation again. So they are developing other mutations in the BTK, but not the cysteine 481S. That actually goes away. It is also shown here graphically where at the time of resistance to LOXO, or it's called pirtobrutinib now, you are seeing this L528W, T474I, and other mutations. You're not seeing cysteine 481S, which if it was present before starting pirto, kind of goes away. So this is a new kind of field which is evolving, where we are also now starting to see some data that zanubrutinib can lead to some patients achieving having L528W. Acalabrutinib can lead to some patients having T74I mutations, and rarely these have been described with ibrutinib as well. So I think moving forward, I suspect that if you're going to switch from a one BTK to another BTK, you have to know the resistance pattern, because if you have a patient, let's suppose the patient you treated with zanubrutinib, and the patient has developed an L528W mutation as a resistant to zanubrutinib, then using pirtobrutinib is not going to work. So I think this is a similar story to obviously with CML, where we know it's a very common thing, obviously, to check, and it's an important thing to check whether a patient has a T315I mutation or not. I think so. The kind of the CML kind of a story with ABL resistance mutations, I think is going to start to play out in the context of CLL with BTK as well. Now, besides pirtobrutinib, which is a very important drug in the field and likely to be FDA approved for CLLs hopefully soon, the other important drug, or not drug, strategy is CAR T-cell therapy, which was recently presented at the ASCO meeting by Tanya Siddiqui. So this is a Transcend CLL 004 study. The CAR T is lysocaptogen maralaclucil, or also called Lysocell. So these patients, relapsed refractory CLL, it is an autologous scar, so they got leukapheresis, then they got flu-cy lymphodepletion, and then they got CAR T at two different dose levels, 50 million total cells or 100 million total cells. Total 117 patients were treated, so a large CAR T study, 117 CLL patients were treated. They also looked at the subset of patients who were BTK progression and venetoclax failure, which was about 70 patients. Now, and then they had their subset was about the 70 patients who had both BTK progression and failure. They looked at the patients who received dose level 2, which is their target dose, 100 million cells, which was 49 patients. If you look at the response rate, CR/CRI, it was about 18%. So maybe numerically it appears to be on the lower side. If you look at the MRD rates in the blood, it was 63%. MRD rate in the bone marrow was 59%. So I think about, MRDs tend to be more important in CLL. We are realizing compared to CR/CRI rate. So I think, half or slightly more than half of the patients will achieve MRD negative remission in relapsed refractory CLL who get this CAR T. And when you look at the progression-free survival for the entire patient population or this specifically identified group of patients, which they kind of said as efficacy evaluable, you can see the median progression-free survival was about 12 months for this group of patients who were double kind of exposed/refractory. Entire population was about 18 months. So certainly, I think these patients are achieving remission, but you can see these patients are relapsing. We don't really see a tail to the curve right now. Obviously, a longer follow-up trial of this will be very important to see what happens to these patients who are now post 1 year of follow-up. So these two strategies I mentioned to you, one is pirtobrutinib, one is liso-cel. I kind of, in one slide, I'm going to show you some of the major data, because I think these two are going to become very important strategies for these double exposed, double refractory patients, where patients who have failed covalent BTK, have failed venetoclax, and they're coming to clinical practice for trial options. So pirtobrutinib, I showed you the data for this double exposed patients. The median is about 17 months. For this liso-cel, again, slightly smaller number of patients. It's about 11 months in terms of the treatment approach. Now, in the last maybe couple of minutes, there are additional novel agents in CLL which are coming along. So I mentioned before, there is a BCL2 inhibitor called esaftoclax. It was called APG-2575 previously. It's by a company called Ascentage. There's also sonrotoclax, which used to be called BGB-11417, which is by a company called BeiGene. So these two are going in phase 3 clinical trials right now in CLL. So we'll see how that kind of pans out. There's also already reported data in a phase 1, phase 2 study. There are other non-covalent BTK inhibitors which are in clinical development. At least these are the ones I know because I've been involved in some fashion. Carnabio is a currently ongoing study at MD Anderson and a few other places. And there are other companies which have these novel non-covalent BTK inhibitors. So these are pirtobrutinib or nemtabrutinib-like drugs, which is in early phase trials. There is a PKC beta inhibitor. PKC beta enzyme is one step downstream of BTK, and a company has an oral drug which we are part of. There are BTK degraders, which are actually becoming quite an important concept. Multiple companies have BTK degraders now, which degrade the entire BTK protein. And CD20 bispecifics are in clinical development. Early phase trials are ongoing. Obviously, CD20 bispecifics have made a big splash in DLBCL and lymphomas, but for CLL, the data has lagged behind. My last slide here, then in conclusion, I think hopefully I showed you for the frontline CLL, I think we already have effective therapies, including BTK, BCL2 CD20 antibody, BCL2 BTK. So for example, ibrutinib plus venetoclax approved in the EU, but not in the US. And the triplets are undergoing randomized studies, and we'll see how that pans out. For the relapsed CLL, I think the treatment choice really depends on the type of treatment patient has received before. So it's very important to get that history out. Non-covalent BTK inhibitors really looks promising. We have this data with CAR T I showed you. I think that data is being evaluated by the FDA for approval. We'll see how that pans out. But there are all these other novel targets which are in pipeline as well. We'll have to see how that kind of pans out, how the phase 1, phase 2 studies get, the data we see. I did not talk about Richter's transformation, but that remains an unmet medical need. There are several agents in development, including venetoclax, pirtobrutinib, checkpoints, which we have participated as well as CAR T-cell therapy. So that remains an unmet need, and hopefully we'll wait for data on these strategies for these patients. With this, I would like to conclude and happy to take any questions.